Researchers have had a breakthrough in the central questions about the origin of life. A collaboration of physicists and biologists in Germany has found a simple mechanism that might have enabled liquid droplets to evolve into living cells in early Earth's primordial soup. How did these first cells arise from primitive precursors? Dubbed protocells, how did these precursors come alive? How could something as complex as a membrane start to self-replicate and proliferate, allowing evolution to act on it? New work by David Zwicker along with the Max Planck Institute for the Physics of Complex Systems and the Max Planck Institute of Molecular Cell Biology and Genetics, both in Dresden, may have an answer. The scientists studied the physics of chemically active droplets which cycle chemicals in and out of the surrounding fluid, and discovered these droplets tend to grow to cell size and divide just like cells, as well as move across cells via diffusion. This active droplet behavior is different from more familiar tendencies of oil droplets in water, which glom together into bigger and bigger droplets without dividing. Frank Ulicker, a biophysicist in Dresden and co-author of the new paper, said, "It makes it more plausible that there could have been spontaneous emergence of life from non-living soup. Is it possible that these chemically active droplets are responsible for the evolution of modern life for everything from amoeba to zebras? Experiments are underway to observe the growth and division of active droplets made of synthetic polymers that are modeled after the droplets found in living cells." Frank and his team are not the only ones interested in this topic. David Deamer, a biochemist at the University of California, Santa Cruz, and a longtime champion of the membrane first hypothesis, admits that while droplet division is interesting, he contends its relevance to the origin of life remains to be seen. Deamer believes that the multi-step process by which modern cells divide is far different from the simplicity of chemically charged droplets. Princeton University's biophysicist Clifford Brengwin, who was part of the Dresden-based study, explained that it would not be surprising if these were vestiges of evolutionary history, like mitochondria. Organelles that have their own DNA came from ancient bacteria that infected cells and developed a symbiotic relationship with them. The condensed liquid phases that we see in living cells might reflect, in a similar sense, a sort of fossil record of the driving forces that helped set up cells in the first place, he said.